Hello guys, welcome to my YouTube channel Biotech-India. Today I will be covering a topic of plasma membrane. So in this we'll see where do you find the plasma membrane in the cell, what is a semi-permeable property, how about its structure, and what all different types of membrane proteins which are present on plasma membrane and its function. Membranes are present around the cell and each of the cell organelles. It separates cellular materials from external environment. It also regulates which material can enter and exits in the cells, like how the security guard stops and asks you an information before entering into an apartment or club. It also maintains homeostasis in the cells. So homeostasis is nothing but a self-regulating process by which biological system maintains stability while adjusting to changing external conditions. What about the semi-permeable property? So a membrane that allows certain material to pass through based on certain properties, size, its hydrophobicity, charge, etc. So basically it's a barrier, or as a sieving, it acts as a sieving. So there are several different models which were proposed to explain the structure of plasma membrane. So in 1935, Daniel and Dyson proposed the sandwich model. In this model, a lipid bilayer which was coated on either sides of the protein. Okay. So mutual attraction between the hydrocarbon chains of lipid and electrostatic force between the protein and the head of the lipid was thought to maintain its stability of the membrane. In 1960, Robertson proposed a unit membrane model. So it says all cellular membranes have identical trilaminar structure, that is dark, light, dark. In 1972, S. Jonathan Singer and Grath Nicholson developed a new ideas for membrane structure. They proposed a fluid mosaic model, which is dominant model now. According to this model, it has two key features: cell membranes which contain bimolecular layer both surfaces of which are interrupted by protein molecules, and proteins occurs in the form of globular molecules and they are dotted about here and there in the mosaic pattern. As you can see here, this is a lipid bilayer and these are the proteins which are embedded here and there in the mosaic form. Okay. The lipid bilayer suggestions agrees the previous models, but views of protein as a globular entities embedded in the layer instead of the thin sheet on the surface. Coming towards its structure, so basically there are three components: carbohydrates, lipids, and membrane proteins. In 1962, Bell suggested the carbohydrates in the plasma membrane. So these are located extensively on ectoplasmic surface basically. They play a very important role not only in the mechanical structure of membrane but also as a carriers or channels serving for transport. These are short, unbranched, or branched chain of sugar attached either to exterior ectoproteins or polar ends of phospholipids at the external surface of plasma membrane, which forms glycoproteins and glycolipids respectively. Membrane carbohydrates occurs as a glycoproteins and glycolipids. Most of the membrane carbohydrates are bound to protein molecule. Coming towards structure of lipid component, there are four major classes of lipids, which are phospholipids, sphingolipids, glycolipids, and sterols. So these are amphipathic in nature, which have hydrophilic head and hydrophobic tails of unsaturated fatty acids. So, thanks to the amphipathic nature of lipids, in water or aqueous solution, phospholipids tend to arrange themselves in these structures. So basically, if it has, so they have a hydrophobic head which are outside and the tail which is inside. A hydrophobic tail which is inside. If the tails are small, they form a myceli, and if they are bulkier, they form a liposome, which is a hollow droplet of bilayer membrane. Coming to a different types of membrane protein, first are integral proteins, which are immersed in bilayer and held in place by affinity of hydrophobic parts of the protein and hydrophobic tails of phospholipids on the interior of the layer. So they are permanently embedded in plasma membrane. They have functions for they have different functions which includes the channeling and transporting molecules across the membrane. As you can see in the figure, there are two different types of membrane proteins which are associated molecules, group 1 and 2 transmembrane proteins which are in opposite orientation. Another is peripheral proteins. These are the temporarily associated proteins, and these are more hydrophilic in natures and thus are non-covalently linked to a polar heads of phospholipids and other hydrophilic parts of other membrane proteins on surface of the membrane. So these also can be attached to integral membrane protein. These are often associated with ion channels and transmembrane receptors. Another type of proteins are lipid anchored proteins, essentially hydrophilic, so are associated so are located on the surface of membrane and are covalently attached to lipid molecule embedded in the layer. There are other different types of membrane proteins which are present. First is cell-cell recognition protein. Carbohydrate chains of glycoprotein and glycolipid serves as a fingerprint of the cell and identify the type of cell. So glycoproteins on extracellular surface serves as a ID tags which recognize individual different species, different types of cells. Second is integrins, which provide strength to the membrane. These integrins are attached to cytoskeletons and other extracellular matrix to maintain the cell shape and stabilize proteins. Third are intracellular junction proteins. These proteins help cells to stick together to form tissues. It binds cells together through tight and gap junctions. So in tight junction, it seals cells together to form sheet to provide an impermeable barrier to the cell membrane. Also, it prevents transfer of water and water soluble substances through the membrane. While in gap junctions, they provide direct connection between the cells. They are some sort of channels or pores through the membrane of cells and across the intracellular space. Fourth are enzymatic membrane proteins. These proteins are attached to integral membrane proteins or penetrate the peripheral regions of lipid bilayer. The regulatory proteins, subunit of many ion channels and transmembrane receptors, may be as a peripheral membrane proteins. Examples are G proteins and few of the protein kinases. Signal transduction protein, these are the receptor proteins, bind hormones and other substances on the outside of the cell membrane, which triggers a change inside the cell, which is called signal transduction. For example, the binding of insulin to insulin receptors causes the to put glucose transport protein into the membrane. Sixth is the transport proteins. These are responsible for passive and active transport. So in passive transport, the transport is happening from high concentration to low. So there is no need of energy required and the equilibrium is reached. While in active transport, the transport is from low concentration gradient to high concentration gradient. So it requires energy in the form of ATP and the concentrations become unbalanced. So in my upcoming video, I'll be describing more detail about uh, the membrane transport proteins. So stay tuned. Thank you very much.